Thanks a lot, Jonathan. Merci, Jonathan. Next up, we have Dr. André Nantel with A Caveman, His iPhone, and the Rise of Systems Biology. Ensuite, je vous présente Dr. André Nantel.
So, hi, my name is André. I'm a biologist at the National Research Council of Canada. I'm also an adjunct professor at McGill University. I'm also known for publishing beautiful pictures on the internet. Part of my research is to try to figure out and understand how you can start with a... I take that into account. How can you start with a chemical code that's actually smaller than an MP3 file and end up with something like serapalin? So to understand that, you need to know the first tenet in biology. Cells are the basic units of life. They're wonderful little machines that also have a very developed information processing apparatus. And although it's much simpler than and less versatile than a living cell, most biologists would be really hard-pressed to figure out a simple iPhone, how it works. And very often in my job, I'm confronted with problems that are so complex, so infinitely detailed, that I feel like a caveman who's just found one of those and is trying to figure out how it works. How do you do that? Well, you use a rock or a blender, and you separate it into its individual components, and you see how they interact with each other. In my world, that's the job of a biochemist. Now, the first half of the 20th century, that was the golden era of biochemistry. We have found that we are ugly bags of mostly water, but also composed of nucleic acid, proteins, carbohydrates, and lipids. Now, we also figured out the transmission of biological information in 1953, where these two guys stole her data to figure out the structure of DNA. And one of them went on to discover what is the genetic code, leading to the era of molecular biology and the dogma of molecular biology, which is essentially genes are in DNA, which is transcribed into a similar molecule that's called a messenger RNA. The messenger RNA is read 3 bases at a time into one of 20 amino acids that make proteins, and protein make everything else. So say we all. Over the next 50 years, molecular biologists had a lot of fun splicing DNA around and making a bunch of disgusting mutants that taught us a lot of things on biological systems. But molecular biology is also a very semi-quantitative science. Most of our results are done by looking at gels and is the band there and how intense it is. And phrases like the one on top were fairly common at the beginning of my career, unfortunately. Now, this is because biologists are really good at taking something very complex and simplifying it, extracting its simplest entity. This is how a cell looks like in the Bible of molecular biology. This is how it looks like in reality. Now, the world is changing. The human genome came out in 2003, and our ability to produce genomic data has been following Moore's law and growing at an exponential rate. That has had a massive impact on how we do things in the lab. For example, this is my first scientific paper published almost 20 years ago. It took me 2 years to clone, sequence, and characterize one gene from canola. Now I could do this in 2 weeks, working 6 hours a day, taking weekends off. I'm in government, what can you say? Here's a gold standard. During the cholera epidemic in Haiti, researchers were able to sequence the entire genome of the responsible bacteria in 90 minutes. That's shorter than the Ignite. And to figure out what it means in 2 days. Here's another example from my own lab. We're using a technology called DNA microarrays to read the expression level of the 40,000 genes in human cells. And we use that, for example, to look at various pediatric brain tumors and see how different they are from each other, even though they look the same. And now we're able to, we know a lot how cells sense their environment and respond to these changes. But even with all of that, our knowledge is still very limited. Here's a similarly detailed pathway on the right, and you know a lot is missing. So now we have so much data, so much information, so much relationship between them that it's becoming impossible to simply describe them in simple terms. And you're seeing a fusion of biology, mathematics, and bioinformatics into a whole new field that's called systems biology. And systems biologists will take data from a variety of sources, organizing them in machine-readable form that actually look very much like computer circuits, electronic circuits. And just like the weather satellite gave the meteorologists tons and tons of data that they were able to use to build climate model. It's the same thing in biology. We're using these data to build biological systems model. And for us to be able to do this successfully, God forbid, we're going to need engineers.